and get found dead in the trunk. Fake the funk and get found dead in the trunk. Fake the funk and get found dead in the trunk. Fake the funk and get found dead.
in real-time computing. Okay, without further ado, not stop. Today we are very kind of honored to have Dr. Saket Navlakha. He has very limited position. Currently, there are 3 Nobel Prize winners at the Saket Institute. I told him maybe he will be the next one. Okay, so Saket research interests include design algorithms to study the structure and the function of biology network and the study of algorithm in nature. Recently, he had one paper got accepted by Science. Science is probably the best journal in science. So in the first part, you will talk about the work of the Science journal work. Okay, so welcome. Yeah, thank you.
Thank you, Shuqing, for the introduction, and it's always exciting to be here, but intimidating for many reasons as well. But let's see how this goes. So I want to tell you 2 short stories today about some work we've been doing the last couple of years at the Salk, and both of these stories are the basis of them is to think about biology in terms of how these systems have evolved to solve computational problems that we think about a lot in computer science. And the goal is to both use the language of algorithms to kind of understand how these systems are solving these problems, but then also hopefully get some inspiration from them to develop new algorithms. Okay, so the first story I want to get right into is a problem on similarity search or nearest neighbor search, and I want to talk about how the brain approaches this problem. And so this is joint work with Sanjoy Dasgupta at UCSD and my colleague Chuck Stevens, a neuroscientist at the Salk Institute. Okay, so here's the problem. This is something that we can do remarkably well in our brains. You look at somebody and you're like, oh, that guy reminds me of Mark. Or you hear a song and you're like, oh, I've heard that song, or I know this band before. Or you smell something and you're like, oh, that smell reminds me of a certain fruit or an orange. Right? So think about what's happening there. I have a whole potential lifetime of experiences of people I've seen or things I've smelled or music I've heard. And then I have this query that's coming at me in real time. And somehow I need to search this database that I've encoded in my brain of all these experiences that I've had to determine, should I go up and say hello to this guy? Or should I change the radio station? Or should I not eat this because I might be allergic to it? Right? So that somehow we're doing this so efficiently and so fast, we're performing the similarity search. And so we got thinking about how is the brain solving this problem? And from a computer science perspective, this is something that you're very familiar with as a problem that many information retrieval and information technology companies have to solve. So for example, Pandora, you put in a song and it tells you, here's a whole bunch of songs in our database that are similar that you might also like to this song. Or you watch a video on YouTube and it's going to say, here are a whole bunch of videos that are similar to the one that you're watching. Or you go to Amazon and you're searching for a product and they're saying, okay, here are other products that you might be interested in. Or Google does this for duplication detection. It looks for similar documents on the web that might be duplicates of each other. Okay, so this is a same fundamental problem, right? I have some query, I have a large database of things I've seen in the past, and somehow I want to do an efficient similarity search to determine how to deal with this product. So my goal today is to do 2 things. One is to tell you a common computer scientist approach to solve these problems that are very practical and used today in a lot of these cases. And then second is I want to tell you about how the fruit fly brain, this little fruit fly, solves a very similar version of this problem, and then kind of do a little bake-off and see how they do against each other. Okay? Okay, so here's the computer scientist problem. We're going to think about it as image similarity search, but it's all the same idea. So I have a query, some image, and then I have a huge database, billions of images that I've seen before. And all I want to do is I want to search through this database and find, let's say, the 3 most similar images that look like this guy over here. Okay, so how are you going to solve this problem? What algorithm are you going to use? So often the first thing you do is you represent your image as some d-dimensional feature vector. So these could be the pixel values of the image, it could be some higher level features, however you want to represent an image. You just represent it as a vector in d dimensions. And you can think of this as music or videos, or it doesn't matter what it is, you just have a d-dimensional feature vector. Okay, so now finding its nearest neighbors or its most similar neighbors corresponds to finding other vectors in this d-dimensional space that are very similar to it. Let's say low Euclidean distance between them. Okay, so how would you solve this problem now? So the first thought is, okay, why is this a big deal? Why don't I just do linear search, go one by one through my whole database, and find all the guys that are similar to this? Okay, clearly that will work. You'll find the exact nearest neighbors, but the problem is that it's not going to be a very scalable algorithm, right? You're going to literally have to go one by one through the whole thing, and so if you have a large database, this is not going to be a very useful solution. There's a whole other class of algorithms that I won't talk about today that work much better for large databases, but for relatively low dimensions. So if you look at space partitioning methods or KD trees, you can find the exact nearest neighbors in logarithmic time, but all of these methods only work if your dimension is, let's say, around 10 to 20, but not much larger than that. So the problem has remained is, what do you do if I have a huge database and every element in my database is very high dimensional? Then how do you solve this similarity search problem? And what this has motivated, so about 20, 30 years ago, what this has motivated is this idea of, instead of finding the exact nearest neighbors or the exact most similar items, what if I relax that requirement and say, just give me something that's good enough? Right? In a lot of these applications, you look at an image, you don't need the exact nearest neighbors. As long as it's good enough, you're usually going to be happy with that. So the question then is, if I relax this and say, just give me something good enough, can I solve this problem much more efficiently? And the main algorithm that people have thought about is to use the idea of hashing to solve this problem. So you might have seen hash functions before, where you take some images and you hash them into buckets. But what a conventional hash function is going to do is, if you look at all the images that get put in the same bucket, they're not going to have any notion of similarity to each other. Right? Anything can belong, two random things can have equal probability of belonging to the same bucket as two things that are very far apart. So this is great if you're storing passwords in a database, but if you want to use hashing for similarity search, you would like your hash function to have this property that's called locality sensitive. And what this means is that if two things are similar in input space, there's a higher probability or a higher likelihood that they're going to be placed into the same bucket. Why does this help? Because now I can take my image, figure out what bucket it lies in, and then just look very locally into that bucket and find my nearest neighbors without having to deal with the rest of the database. Okay? So that's what locality-sensitive hashing is compared to what a conventional hash function is. And just to formalize this idea that, again, has been proposed many years ago, is a hash function h takes some d-dimensional vector, your image, and hashes it into usually some lower dimensional space. And this hash function h is called locality-sensitive. If I take 2 points in the input space, the probability that they get assigned the same hash is equal to their similarity in input space. Okay, so the more similar they are in input space, the more likely they're going to get placed into the same bucket. Okay? So, and similarity could be based on, for example, the Euclidean distance between their L2 norm between their vectors, or however you want to define similarity. Okay. So how do you actually, or how have computer scientists gone about actually constructing a hash function with this property? And the main idea is to use random projections of the inputs. So here, let's say, is my input vector x. What we typically do is we want to reduce the dimensionality of this input, but we want to reduce it in such a way that if I have two things that are similar in this input space, the chance that they're going to be similar in this lower dimensional embedding is going to be very high. And the way that you guarantee that is using random projections in something called the Johnson-Lindenstrass lemma, which provides theoretical guarantees that say that if I multiply this d-dimensional vector by a random projection, then these interpoint distances in this low dimensional space are going to be preserved. Okay, so the idea is that I can take any data from any dimensional space and embed it into another dimensional space while still preserving the basic distances between the points. Okay, and so just to formalize that, so I'm going to, let's say I do this a few times, I'm going to take my input vector x, I'm going to multiply it by a Gaussian random variable, and then you have to do some stuff here just to discretize it because we want to deal with integers, and then that becomes my hash corresponding to that, and then I might concatenate multiple hashes together, and now that becomes my representation for that image. Okay, and now given this, if you buy that this works, the way you can solve this problem is very easy. I take my query, I compute h of x, its hash, and then I go into that bucket that it gets assigned into, and then I look what's in there, and then I can just sort these however I want and return the most similar images that I see. Yes?
So this hash function is usually some numbers or just one number?
So it's a parameter of the algorithm. What I've shown here is just one number, but in reality, you're going to concatenate some number of hashes together, and that's going to be the bit number. And so, the higher you increase that, the more you might have to, more tables you might have to create. The lower it is, you might get a lot of noise. So it's a parameter that you have to set. But just for now, I'm just saying, let's just say you have that. Thanks. Okay. Okay, so this is a very core algorithm. There's many bells and whistles on top of this, but a very core algorithm to solve this problem. Okay, so now I want to tell you about fly olfaction and the connection that we made between this locality-sensitive hashing problem and what the fly needs to do. So I want to tell you three things. One is, why might the fly need to perform similarity search in its brain? What is the fly's algorithm to solve this problem? And then we want to compare it to what I just showed you of conventional LSH. Okay, so the goal of the olfactory system, the odor processing part of the fly's brain, is to take some odor that it smells from the environment and assign it what neuroscientists have for a long time called a tag. And for our purposes, the tag is basically going to be a hash. And what the tag corresponds to is a set of neurons that fire whenever that odor is presented or experienced. Okay, and the goal is you want to create some kind of encoding for this odor so that the fly can then learn, oh, do I want to approach and eat this thing, or do I want to run away from it because it might harm me, whatever the case. The first thing it needs to do is identify that, and it does that by generating this tag for the odor. So what we asked is, what is known about the process by which the fly takes something from the environment, smells it, and assigns it this tag? And over the last 5 to 10 years or so, many labs have been working on this problem, and they've discovered this 3-step procedure that I'm going to describe to you now of how you take this odor and compute its hash or its tag in the brain. Okay, so let me describe these 3 steps to you now. Okay, so the first step, these are 50 neurons. They're called odorant receptor neurons in the fly's nose. The names don't matter. 50 neurons. And what these guys do is represent any odor that the fly smells as a point in 50-dimensional space. Right? So there's 50 neurons here for 50 classes, but you can just say there's 50 neurons. And each of them is firing at some rate. And so that defines a point in 50-dimensional space. So we're going to operate here in d equals 50. And these guys, in the first step of the circuit, project to 50 neurons in a one-to-one manner called glomeruli. And as a result of this projection, here's what happens. So if I plot the firing rate distribution of the 50 neurons here, you'll get an exponential distribution, but the mean of that exponential is going to be dependent on the concentration of the odor. So the higher the concentration, the higher the mean. The lower the concentration, the lower the mean. As a result of this projection, this factor gets normalized out, so that if you plot the distribution of firing rates at the glomeruli, they all have an exponential distribution with the same mean. Okay, so what's the big deal here? What the fly is essentially doing is just centering the data, right? We do this all the time. We have a whole bunch of vectors. We want to assign them a similar mean so that we can then compare them in a fair manner. So the fly figured out that this is the first step that it wants to do. So now different neurons could be firing at the tail of this distribution for different odors, but they all have the same distribution that they're going to start with as an encoding. Okay, so this is kind of a technical detail, but we just thought it was kind of cool that the fly figured out that you first need to normalize whatever input you have before you proceed. Okay, so now is where things start to diverge from what I showed you before. So what I showed you before was I have my d-dimensional representation, and then I reduce the dimension and then compute hashes. What the fly does is actually the exact opposite thing. So it takes its 50 glomeruli and projects this 50-dimensional point into a 40-fold larger dimensional space, 2,000 Kenyan cells between these two guys. So it expands the dimension by 40-fold. And amazingly, this connection matrix between the glomeruli and these 2,000 cells called the Kenyan cells is a random projection matrix. Took about many years of work, but they figured out there's no structure in this matrix. But it's not a Gaussian random matrix, it's actually a sparse binary random projection matrix, which is much more efficient to compute than doing a Gaussian, all the Gaussian multiplications and additions that you have to do. So in particular, every Kenyan cell here is going to sample from 6 random glomeruli and sum up their firing rates, and that's going to define its firing rate. Okay, so the main idea, instead of dimensionality reduction, we did this massive dimensionality expansion using a random projection. So now, all of these neurons here, depending on who they're sampling from, are going to be firing at some rate. And then the third step is also very clever, is that all 2,000 of these cells project to this one neuron called APL. It's an inhibitory neuron in the brain. And this guy provides feedback inhibition, and the result of this is that 95% of the cells get shut off. And all I'm left with is 5% of the cells that are still active for that odor, or for whatever that input is. And these 5% correspond to the highest firing 5% that I saw here. Okay, and then this corresponds to the tag that the fly has computed for that odor. So what we've gone from is a low-dimensional, dense representation to a very high-dimensional representation that's very sparse. And the hope now is that because I have these points that are so sparsely distributed in a high-dimensional space, if two guys lie very close in this space, you're pretty sure that they're going to be similar neighbors or very near to one another, which we're going to compare to next. Okay, so just to summarize, the first step is to center the mean, do this massive dimensionality expansion using a random projection matrix, and then sparsify it by leaving only a few cells firing, and that corresponds to the hash. Okay, so the first thing that we did was this way of doing this random projection is unusual, doing sparse binary into an expanded space as opposed to a dense Gaussian. So what we did is we showed theoretically that this form of random projection also satisfies this Johnson-Lindenstrass lemma. And what that means is that if I have two input points x, they have some distance in their original space, I can read off their distance after hashing them by just multiplying it by some constant roughly. So what this means is that after hashing, their expected distances in this expanded space is preserved up to a factor of their original space. So this is just a result about expected distances, but the reality could be very different if the variance is high. And what we showed is that if you do enough random projections, even the variance of the hash is going to be concentrated around its expected value in this 1 plus or minus epsilon manner, so that you get a very tight bound in terms of its distance preserving properties. So these two lemmas are basically stating that this form of random projection that the fly discovered also satisfies this idea of distance preservation and can be used as another type of hash function. Okay, so then we compared these two approaches. So we took the standard approach, which takes some d-dimensional vector and does a dimensionality reduction. And here we did it, we compared them in such a way that we kept the number of mathematical operations required the same. So every time you do a Gaussian random projection, you have to do d multiplications and then d additions to compute the hash. So it requires 2d operations, and then you do this some number k times. Okay, when you do a sparse binary random projection, the number of operations is much lower. So you're sampling with 6 from 50, so it's about 10%. So you're saving 20-fold on the number of operations, and so we said, okay, we're going to do just 20k the number of random projections into this expanded space. And then we also have this additional step of sparsification, but that's pretty easy to compute. But the idea is that we're going to compare both of these by keeping the number of operations that both have to perform to be exactly the same. Okay? So we took some benchmark data sets. Again, so this problem has been studied for many years. And so there's a lot of benchmark data sets for doing nearest neighbor search. So we took 3 data sets, all with very high dimensions such that KD trees and these other kind of things won't work. And we did exactly what the problem states. So we took a query image. We found its top 2% of its true nearest neighbors in d-dimensional input space. Then we computed their hashes, and we looked at their top 2% of predicted nearest neighbors in hash space. And then we said, okay, what is the overlap between the true and the predicted nearest neighbors? And that defines its precision. And we compared them by varying the hash length, which is this number of non-zero entries that are going to be used in the hash representation. Okay, so here was the first thing we did is we asked what happens if you just replace this very expensive dense Gaussian random projection with a more efficient sparse binary one. And so what we showed, so on the x-axis here is the hash length, the number of non-zero entries, and the y-axis here is the overlap of true and predicted nearest neighbors, so higher is better. And what we're showing here is that by reducing the complexity to do a sparse binary, you're essentially losing no information in terms of identifying nearest neighbors. So both of these lines are almost exactly overlapping. But the main advantage here is that I've gained a factor of 20 in terms of the efficiency of computing these in these hashes. So that's the first step. Then we said, okay, what about the second step of doing this massive expansion versus the dimensionality reduction? And so again, same plots here, hash length versus overlap. The red is what you would get if you don't do any expansion. The blue is what happens when you actually do the expansion and then do this winner-take-all circuit to identify the highest firing neurons. And so we could see throughout the entire hash length, we see this boost in performance over what you would get from standard locality-sensitive hashing. And then finally, we implemented the full FLIES algorithm where we did an even larger expansion, a 10-fold expansion. And so you see the similar trend. The blue here is the FLIES algorithm. The red is LSH. And especially at these very small hash lengths, we see improvement about 2 to 3-fold, and not as much in the larger hash lengths, but mostly in the smaller hash lengths. We see quite an advantage in using this FLIES algorithm to identify nearest neighbors. So we were quite excited about this from two perspectives. So one from a computer science perspective, by studying this neural circuit in the brain, we were able to find this new algorithm for locality-sensitive hashing, for efficiently finding nearest neighbors of very high dimensional points. And we've also been excited from a neuroscience perspective, because I think one of the goals of neuroscience is to understand the brain as a set of algorithms. And this idea of thinking about algorithms provides a new way of approaching how is the fly solving this important problem of olfaction. And I motivated this by looking at audio and face similarity search, and now we're looking at the circuits in the brain that are performing these problems, and it looks like it's a very similar idea that it's using there. So this seems to be a very general way of solving this kind of computational problem. Okay, so that's the end of the first part, and
I have a question.
Yeah.
So when you speak about a fly, and we talk about sensing,
Yeah.
I guess that is sort of inherent to their survivability.
Yes.
So that might be a static algorithm in place.
Yeah.
While if you compare to, say, humans, we do a lot more, and that might be akin to I think it's the system where we don't think, but we reflect on, we do things.
Yeah.
While with humans, we have more computational adaptive mechanisms.
Yeah.
So we might not be as efficient as the fly, but at the same time, maybe we can do more than the
Yeah,
fly.
it's a great point. So I'll tell you something that, for example, something that humans can do that flies can't do. So if you give me 2 types of red wine today, I couldn't tell them apart at all. But if I took classes, if I trained myself and kept smelling and trying it over time, I can start to separate the tags so that I can associate and say, drink this type of wine and not this type of wine. The fly can't do that. It doesn't have the machinery built into that. And so that's called learning, and in computer science we call it data-dependent hashing, where you actually learn from previous data and modify your hash over time to reflect some evolving notion of what's similar and what's different and what I want to discriminate. So what I showed you in this 3-step circuit, in the mouse and in the human, there's a 4th layer of neurons that perform this function. And that's something that we're looking into now to see how you can actually do that in a computational way. So yeah, it's a great point. Yeah, thank you. Yeah.
Really cool stuff. So I have a question in terms of the computational capacity. So when you sort of looking at the number of operations, I mean, obviously you will have like non-human kind of computation operations, how to count those operations in
Yeah.
line. But like TrueFly, they neural network are not processing the same way.
Yeah.
So how do you sort of? I guess it's a general question. I mean, how it's good to have sort of bio-inspired algorithms, but not necessarily actually computationally would win when you sort of perform that on the traditional computers. So
Yeah.
How do you sort of look at those?
Yes, so in our case, we were just trying to come up with a fair way to compare two different algorithms that were being used, and so we decided to look at number of mathematical operations as a good way to level the playing field. But you're right, in the brain, these things, I mean, they do matter because the brain is taking up 30% of our energy. It's the biggest consumer of energy. And these things do matter if you do it at the scale. We have 100 billion neurons in our brains, and this thing adds up. So there is some sense of wanting to do things in an efficient manner, but what the trade-off is between doing it efficiently enough and getting the right performance, I think is a tough question for us to answer right now. Everything is happening in parallel, which also, for our case, that doesn't matter as much, but in this case it does. So I think it's tough to understand what is the right way to compare it in the brain, but we were just kind of thinking about it in a very, as you said, a von Neumann way, and so that made sense to us. But it's a good point. It's not sure what the right way to trade off the two is. Thanks. Yeah.
So, so from the fly scan, brain scan, can we understand what kind of things they have smelled for now? I mean, that is more like neuroscience.
Oh, like what kind of odors are presented to the
yeah,
fly?
so say we don't know what type of odors the fly have smelled so far.
Uh-huh.
We want to scan the brain of the fly, and then we have to detect what have been smelled by this fly before.
Oh, yeah.
Can it be done?
So it's hard to know what the fly has smelt in its past, but you can do experiments where you keep the fly in a cage and then you present odors to it, and in real time you can record from the neurons that are responding to that odor. So that way you can figure out what is its exact response to any odor that you present.
Because from the brain scan you can understand
Yeah.
what it has smelled?
Yeah, so what you do is you basically put electrodes into the live brain of this fly, and then you present the odor, and then those neurons, whichever ones are going to fire for that odor, start to fire, and then you just read that out from the electrode, and it tells you what the representation is. So it's a really difficult experiment to do, and it takes many years, but that's the idea.
But by this I mean from human brains we can also it can be done for life for human brains.
Well, so there's some ethical issues because we can't do it in the live human brain. They do it in some monkeys and mice. They do it all the time. But yeah, I mean that's the idea of using mice and flies and these things.
So I have my last question. I mean for human brains, do we know exactly which neurons are doing this kind of odor smelling, or say other actions, say for storing memory or storing some kind of information? Do we know exactly which neurons are?
So we know the regions that are responsible for a lot of these processes, and because our brains are just pretty much a scaled up version of the mouse brain, a lot of what the mouse is doing, we can kind of by extension say that humans doing, but we have a lot of other circuitry involved, but the idea is to study the mouse brain and the fly brain to get a sense of what we're doing. And so most of the regions that we have in our brains are also found in the mouse brain. So we know, we have a pretty good idea of what region is doing what.
So adding to this question, is it the fly life cycle is very small, it's limited. So is it some part inherited in the DNA itself that it has some memory coming in from there and some genes are already knowing that this database is built? And if that is the case, then it will reduce the time complexity a lot because you already have it done.
So you can actually, they live about a few weeks, 3 weeks. And you can actually teach flies to do interesting things in those 3 weeks. So all of what I've described here is static, but the behavior that is associated with the responses can change. So you can teach it to approach one type and then run away from another. But what we studied was just this algorithm for computing the hash. What it does with the hash is part 2 of the story, which I haven't talked about, but that's a very fly-centric thing for each fly. Yes?
So you've been talking about odors as a sort of a generic thing, but from an evolutionary perspective, food resource odors aren't the same as pheromones, which aren't the same as pheromones. In other words, you can miss a food odor and not die tomorrow. You can miss a mate odor and still be okay for a few days, but if you miss a predator odor, you're dead.
Yeah.
Is there any kind of, is anybody teasing apart those?
So there's a lot of other brain regions like the amygdala that's involved in a lot of the fear responses that get evoked when you have a predator in sight. And a lot of the parts of the olfactory circuit does project to the amygdala as well. And it can immediately bypass any other response that you would have and just say, just run away now. We haven't been studying that. There are a few labs studying that, but I don't know too much about it. So, yeah.
This is incredibly interesting work. Is this algorithm generalized to other neural circuits with receptive fields like vision or touch?
So at least in early vision, no. Because in early vision, you don't have any notion of this random projection. You have a very topographic map where one region of space is being encoded by a certain neuron and its receptor field. So this idea of using this kind of jumbled up way of encoding information, we don't see it everywhere. Like even in taste, like there's very dedicated labeled line neurons for sweetness and sourness and these types of things. So there are other regions that show this. For example, the hippocampus, where memories are stored, show this. The cerebellum shows this, but it's not. It's used in a very specific way, and so when it's used, in which cases, we're still trying to figure out. But yeah. Okay, so I'll go on to part 2 then, and tell you about a problem on network design. Another important problem in computer science that we've been thinking about by studying plant architectures. And so this is joint work with my student Adam Ulas, who used to be a postdoc in Joanne Corey's lab and who's now at Cold Spring Harbor. Okay, so we started thinking about plants from a very engineering-centric perspective. Okay, so think about trying to build a transportation network, like a subway system. And what you want to do is, let's say you want to get them to reach downtown and you have some places in the suburbs that you want to connect to build a network. The question is, how would you build this network, right? Where would you put the lines and how would you connect everything up? So typically when we think about design problems in engineering, you first ask what is the objective of your system and then you build according to the objective. So here's one very reasonable objective that you would like to have, which is to minimize the travel time between the suburbs and downtown. Okay, so if this were my only objective, the optimal network would be very easy, right? Everybody just beelines it straight up. Okay, the problem with this is that it's very expensive to build, right? You have to lay down all this line, money, labor, materials, all these things. So here's another objective that introduces some tension with this one, and that is to minimize the total length or the total cost of your network. Okay, so then you can ask, what is the optimal thing to minimize total length? And so the answer there is also a well-known network. It's called the spanning tree, or even better, the Steiner tree, which allows you to introduce these non-input points or these branch points to connect things up. But the idea now is that I've connected everything up with the minimal possible length. Okay, so here are my 2 objectives. Travel distance, let's call this guy the satellite tree. And here's the one that minimizes total length, and it's called the Steiner tree. Okay, and so what we asked was, so there's clearly a tension, and you want to design one network, so how do you do it? And so what we asked is, how do plants solve this problem? And why is this problem even relevant to plants? So what we kind of reasoned was that think of downtown now as actually the root where the plant starts to emerge from the ground, and think of the suburbs as the leaves. And what does minimized travel distance corresponds to? It's a measure of how efficiently nutrients can be transported through this architecture. Right? So you have water and nutrients and other things that are being accumulated from the ground. You have sugars and carbohydrates that the leaves are generating from photosynthesis, and all of this needs to be shuttled up and down this architecture. Okay? And the other objective of minimizing total length is just a measure of how many resources do I need to actually build this plant to survive in this environment. And so if you believe, and this is not at all clear that these are actually things that the plant cares about, but if you believe that they are, then the question is how well or how does the plant actually make a good trade-off between these two competitors? Okay, so just to formalize the problem for a sec, so let's say as input I'm given n leaf points, so these are my suburbs, and some base point, my downtown point, and 3D spacing, okay? And what I want you to give me back in return is a set of branches or a tree such that I can minimize the travel distance. So this is the minimum over all the points of the distance along the branches from downtown to that suburb. So the sum of the distances from the base to each leaf. And the second objective is to minimize the total length, which is just going to be the sum of all your branch lengths. Okay, so there's a huge space of possible trees that you might need to scan through even if you only had 19 points. So there's exactly 5.48 times 10 to the 21 possible spanning trees that you can use to connect up these 19 points. Okay, so let's think about the structure of this huge space of phenotypes that you could possibly build. Okay, so here's my space of all possible trees, and I'm going to evaluate them according to these two objectives that we've talked about. Okay, it's total length and it's travel distance. So we have two trees that we've come up with so far. The first is the Steiner tree. So this guy has very low total length, but for some of these guys way out in the suburbs here, they're going to have a long way to travel around to get there. So they're going to have a very high travel distance. The satellite tree has a very large cost, but great in terms of its travel distance. Okay, and so another thing we can do is we can say that these two are optimals. So I can no way get better than the satellite on this axis, and there's no way I can get better than the Steiner on this axis, so all of this is infeasible. Okay, and so if you believe that plants actually care about this, then what we hypothesized is that there would be selective pressure from evolution and natural selection to try and drive these costs down as much as possible as a means to survive, as a means to compete better than otherwise. But right, it's not clear, like how close can you possibly get? I mean, it's unlikely that there's even feasibly a tree that reaches this point, but how close can you possibly get? So what we need is some way to interpolate between these two optimals and see what is going on in the space in between, and then see how good these trade-offs are being made. Okay, so what we did is we thought, let's come up with the simplest possible way to combine both of these into a joint objective. So here are our two objectives. And all I'm going to do is just do a linear interpolation between the two. So instead of minimizing just this guy or just this guy, I'm going to say minimize alpha times this objective plus 1 minus alpha times this objective, where alpha is just a parameter that ranges in between 0 and 1. So if alpha equals 0, then this guy goes away and all I care about is the total length. So the Steiner tree is optimal. If alpha equals 1, I have the satellite. And then we came up with an algorithm, a greedy algorithm that generates a tree that near optimally minimizes this joint objective for any alpha in between 0 and 1. And so just to pictorially demonstrate how this algorithm works, kind of inspired by what the actual plant is doing, we take our input points, we build a little stem going to the centroid of the points, and then we literally just greedily in every step connect an unconnected point to the tree that minimizes this objective. Okay, so you might connect this guy, and then that guy, and then that guy, and then as you go, and you can generate these Steiner points along the way that connect to a branch that already exists. Okay, and so just to give you a sense of what kind of architectures that we can get, so here's actual input points for a plant. I'll tell you how we got this data in a second. Here's the satellite tree that I mentioned. Here's the optimal Steiner tree for these points. Here's what the plant actually looked like. And here are different architectures that are algorithm generated. So this is alpha equals 0.25. It looks closer to the satellite. And as you get to the other extreme, alpha equals 0.75, you get closer to the Steiner tree. And so you can see as you change this value of alpha, you're getting very different architectures. Okay, so what you can do then is use this to generate what people often call in engineering and economics the Pareto front between two objectives. And the idea is that if all these points along this Pareto front, so this is varying alpha from 0 to 1, all these points are not dominated by any other point. In other words, if I improve along one dimension, then that means I suffer along a different dimension. Okay, so what this corresponds to is an optimal trade-off. And maybe in one environment you want to do something over here, maybe in a different environment you want something here, or maybe one species is doing something here and another species is doing something here, but you can kind of tune that the way you want to make this trade-off along this Pareto front. Okay, so just to summarize, our hypothesis then is if plants actually do care about this, then they should be somewhat close to this Pareto front. And the reason we thought this was somewhat interesting to even try was because plants have been around for billions of years, so evolution has had a lot of time to kind of optimize. And they're one of the most genetically diverse species known on the planet. So there's, again, a lot of time and a lot of search space that they can go through to try and optimize this. Okay, so this is the theory. And so how do we test the theory? So what we've been doing over the last few years is doing 3D scanning of plant architectures. So what you see here is a 3D scanner, and this is built by a company called FAIRO. And we called them up, and so their number one client is Boeing, because Boeing uses them for 3D printing and then using them for airplane parts. And so we called them up, and they came by, and we asked them, can we scan plants with this? And they'd never heard of this before, but what we did it, and so this is the scanner. It's manual, so that's Adam's arm right there, and he's going to go around and basically paint the plant with the scanner. And you get a 3D representation in real time. So here's an example of a tomato plant. And the resolution, you can't see it here, but you can even see like the venation patterns in the leaves you get. And no image subtraction, no background, anything. You just get raw 3D cloud points of the exact material of the plant. So it's a really nice way to actually capture this data. You don't even have to touch the plant, so you can do.
How much of the equipment?
75,000.
75,000.
Yeah. So, but yeah, the kind of resolution is.
You
really
mean
nice.
you can capture the texture?
You can capture the texture. Yeah, let me run this thing again. You can see. Oops. As usual, it's better on the screen, but you can, like, all these little lines that you see here, if you look very close to the leaf, that's the actual veins that are going along the leaf to provide transport, and so you can see.
So maybe even this equipment can get like a building, a car?
No, so you're limited to the range of this arm. So basically you can take that arm, which is about 7 feet, and you can scan in that 7 feet range, but yeah, you can't do they have scanners for doing buildings and things, but we didn't need that here. So
that's on my Christmas wish list.
It's a nice toy, yeah. Okay, so this was the technology, and so we basically just grew a whole bunch of plants and we scanned them. So we planted them, we moved them in these very controlled environments, growth chambers where we can control the temperature, control the conditions, and then we scanned them for 20 days, every day for 20 days. And so here, for example, is a tomato plant, and here's the scan version, and this is from the top down. And all we did is exactly what I described. I took the tomato plant, Adam clicked on the point corresponding to downtown or the base, and he clicked on all the points, and we got the XYZs of the locations of where all the leaves are on this, and then we did the analysis from there. Okay, so here's what I'm going to show you a whole bunch of plots that look like this. So this is what I was showing you, the total length and the travel distance. And so this is this tomato plant. We just put it in kind of ambient, very controlled conditions for now. And this is on day 4. Just pick some random days here. And so here we go. So the plant, there we go, right on the Pareto front. So that's the Steiner tree. That's the satellite. This is our algorithm for different values of alpha. And the plant is right there hugging the Pareto front. And just to show that this is like not to happen by chance, we tried out random architectures where you just build random spanning trees. We did this other, not important. But the probability of this happening just by chance was very low. Okay, so we were happy. Day 4, we got something, but is this something that maybe they figured out very early, but then they lose it? But that's not the case at all. So on day 12, it's still right there. On day 20, it's still right there. And kind of we stopped a little bit after that, but throughout its growth period, it's changing where it's lying, but it's still hugging this Pareto front.
So what are the control conditions here? Like, how do you control the resources? Or like a particular amount of fertilizer where the light is coming from?
Yeah, so we did a very basic experiment where we watered it every day. We had top-down lighting, 16 hours, and then 8 hours of darkness, and just normal soil. We weren't kind of playing with all these other parameters. We just wanted to see, in the baseline conditions, is this even worth doing? So, yeah.
So plants take a lot of their nutrients from their roots. Like, if you only look above the soil, you get grass, I guess. But what happens if you don't consider the top and you just consider the bottom, you just consider the roots? Are they both minimizing the total negative nutrient?
Yeah, it's a great question. The problem is that the roots are really hard to scan, because they're underground, and to actually get a nice measurement, you have to basically break them and you lose all their structure.
Can you repeat the question?
Oh, sorry. So the question was, can we do the same analysis on the roots underground as we're doing above ground? Because you would imagine that these two might be important objective for the roots in just the same way that we're showing them upstairs. We want to try it, and there's some technology now where you can grow these in kind of suspended media, where you can actually look at how the roots are growing in the suspended 3D media and then do scanning. But we don't have that technology, and it's still being worked out. But I mean, we're hoping to try it out in the future.
Yeah, because I was reading a paper the other day and using MRI. MRI scans.
Oh, I see.
And then if you use an MRI or a CAT scan, then you can see them.
Oh, I see. Okay. I haven't seen that, but if you don't mind sending me, I'd love to take a look. Thank you.
Or you can use actually like different species of plant, like water hyacinth, which actually floats on the water and grows like that, like lotus flower. And then you can technically just,
I see,
just need water.
Yeah, but you still have to capture the roots underground.
So it could be a transparent beaker, and you float the water in it. And then, I mean,
it could be, yeah. I mean, you would have to do like some kind of image reconstruction, but it's doable. I see what you're saying. Yeah.
So you did this while the plant was growing.
Yeah.
Have you tried it while it was dying?
No, we have.
Understand all of these rules might or they might be kept.
It's a fair point. Yeah. So the longest we went, I won't show data of it, but we went up to day 35, and some fruit was starting to form at that point. But then we stopped scanning after that. Yeah, it's possible that once the fruit is made, it's like, I'm done. I've done my job, and I don't really care about this stuff anymore. But yeah, we don't know yet. So, yeah.
So for the fruits and flies, the competition occurs in the brain. So what are the skills, and they don't have any brain. So
Yeah.
this computation occurs for the brain.
Yeah. Okay. So I'm going to talk about that at the end, but all I'm going to say is I don't know. So, I mean, everything I'm analyzing here is kind of in retrospect. Like, I'm given these points, and now how optimally did I connect them? But you could think that the plant is actually solving a more difficult problem, because it also needs to grow while kind of maintaining this invariant, as we're showing over time. And not only that, but it needs to place these leaves in such a way that it's capturing sunlight. And not only that, but it has to do it in such a way that the competition is not interfering with where it's putting out leaves. So the kind of problem that the actual plant is solving is still very open in our mind, but this was kind of our first step just to see how well it's actually doing, and now we're thinking about, okay, what is its actual strategy?
Competitions are occurring in the cell, or they don't have any brain, right? They don't have neurons.
Yeah, they don't, but the brain is not the only thing that can compute. I mean, there's signaling pathways there's auxin being transported up and down. There's a lot of other forms of information and feedback that is being used to determine, is this a good position? Am I getting eaten by predators? Should I change my strategy? So I think in general in biology, you don't need a brain just to compute. There's a lot of other forms of computation that are happening. But it's a good question. We don't know how all that stuff works. Okay, so we went to the plant biologist and we were like, wow, this is amazing. And they were like, well, you did this in tomato plants in really controlled conditions, but in reality, plants have to thrive in many different conditions. They can't just get up and move. They have to deal with any change to the environment. So we said, okay, if this is a good theory, then we should apply different environments to these tomato plants and see. And what you can see is that all the architectures are changing depending on their environment. So for example, in shade, it thinks that the plant is getting shaded by neighbors, So it shoots up its its hypocaudal to try to get above that shading canopy. In drought, it does other things. In highlight, you can see, so this, I don't know if you can see this, but these leaves turn purplish. It's kind of a sunscreen that leaves use to kind of protect themselves. So there's all kinds of changes in these architectures depending on the environment in which these plants lie. And we quantified some of these things. So for example, the number of leaves, you can get up to 20 leaves versus 5 leaves in different conditions. And how tall it gets really tall over here, but not so tall here. Its total convex hull area or volume can change by orders of magnitude. So there's a lot of plasticity in these plants. And so we said, okay, does our theory still apply to all these different changes as well? Okay, and what we found, so this is shade, again, same type of plots, day 13, day 21. The plant is still, it's changing where it's going, but it's still right there on the parietal front. Then you look at, in the drought condition, it's still hanging out around there. In highlight condition, throughout its development, it's still hanging out around there. And in summary, these are over the 20 days, and we quantified how far the plant is to this optimal parietal front. And in all these cases, it's within 3 to 4% of being right there. Okay, and then, so again, we went back to the plant biologists, and they were like, this is fine, but there's thousands and thousands of plant species, and you only showed it on one species. So we said, okay, let's do more experiments. And we grew this grass type, the sorghum plant that's used. And we did it across 4 conditions. And I won't go through the details, but in a lot of these conditions, you're still seeing the same type of behavior. Interestingly, in highlight, this very stressful condition where you expose these plants to really high frequency light, there is some deviation, and it seems at this point some other priority is happening that's taking over just these 2 objectives. So this is not the whole story, but we're still capturing a lot of the variation here. So just the summary side. So another thing, this is the final thing that I'll talk about, is I talked about, okay, it's nice that they're on the parietal front, but is there any logic to where they lie on the front, right? I mean, maybe they lie here or here or here. This should be telling us that, okay, maybe in this case they're prioritizing this objective, but in this case they're prioritizing a different objective. And are we seeing any structure in that part? And what we found is it was actually quite interesting. So most of the tomatoes, so this is just a mock diagram, but you get the idea. Now, most of the tomato plants were kind of making this 50-50 split between these two, but these sorghums that look more like these grasses were much more pushed towards this side of the parietal front. So, depending on the species, you're trading off what you have. And even cooler to us was we looked at just the tomato plants, and we looked at the different conditions, right? We did these 5 conditions, and what we found was that a lot of them clustered together, but if you put them in high heat, they all jump a little bit more to this side. So somehow in high heat, and this was true in the other species as well, they start to change their strategy to prioritize travel distance. And what's the meaning behind that? It's tough to say, but it's just nice to see that this very simple engineering idea is capturing a lot of the variation that we're seeing in these plant structures. Okay, so I mean, what we're showing, and coming back to your question, is what's the computer science contribution here? It's still to be determined. We still really want to get at what is the actual algorithm that this plant is using as it's growing to maintain this design. And what we're trying to do now is kind of gladiator-style plant experiments where we have 2 in the middle in a little arena, and we want to see how they compete and cooperate in these kind of things for light, and see if these things are still being maintained. From a plant biology perspective, I think this was a nice way of showing that these very general principles of network design are also kind of sculpting the kind of architectures that we're seeing in nature and other species as well. So I just want to thank, so kind of the big theme here again is to think about a lot of these biological processes in terms of their algorithmic properties, and this extends beyond the brain and into other systems as well. And so these are our collaborators on the projects, and happy to take questions.
So with respect to plant networks, since we're in South Florida and we had a hurricane this year, so I was wondering about, another dimension, presumably something about structural strength or ability to survive windstorms or something like that. And it would seem as though the Steiner network would tend to be better for that than the other, but it's not so clear.
Yeah, so the one thing that that was reviewer number 2's comment. And so to address reviewer number 2, we built an architecture that was just structural stability. And so the way, is this the best way? I don't know, but it's a reasonable way, is that we had kind of a palm tree-like, where we have a branch to the centroid and then kind of equal weight dispersion on all sides. And what we found, if you were following the green triangles, which I didn't point to, but they at least alone were not sufficient to dictate what the architecture was. But you're totally right that, you know, we've done these on very small plants, but maybe when you reach a certain height, then this becomes a more important factor. It's not sure, but at least in this early developmental phase for these, we're not seeing that as the dominant factor. But another follow-up to that is everything that we do, everything that I talked about here was length measurements, kind of a skeleton. But we're also seeing, you know, the thickness of the stem increases 4-fold if you put it in highlight. So clearly that's another area of optimization that is important that we didn't take into account here, and that could also help with stability. So, yeah.
Yeah, that was really, really interesting, and I guess I didn't know it was going to be on plants as a plant biologist myself. And I think my background is in genetics, so I'm very familiar with Joanne Corey's work. And I was wondering, on the stress tolerance, are you looking at like genetic networks that are turned on and trying to make correlations? Because that could be really cool, why they kind of in terms of stress.
So that's where Joanne's pushing us next. And so what we're doing now is we're doing mutants. So she's been studying all kinds of light-sensing mutants and other branching mutants. And we're doing this in Arabidopsis because it's just easier to do the mutants there. And so we've collected about 35 different branching architecture mutants, and we're just growing them all out. We're scanning them, and we're seeing. So the cool thing for me there would be to see if we could find a mutant that really pushes this guy off the pareto front. Because then you're saying that gene might be important to actually making this trade-off. And so we're still too early right now to do that, but yeah, that's exactly where we're going next. Thanks.
So that was my question in terms of the relation with genetic material. Because I'm sure some of this stuff must be encoded to be passed on. So I thought that was
Yeah, I mean, it's the natural next thing. We've seen that what it's doing, but now what's the mechanism that's driving it? It's cool. So.
Any more questions?
Did you investigate how to apply this on other real-world applications? For example, like the first example you mentioned, the streets, based on different conditions, environmental conditions.
So actually, one thing that we're doing right now, kind of as a side project, is we're testing if this is even true in cities. So we're basically got 101 cities chosen randomly throughout the world, and from querying Google Maps, we can get the location of downtown. We put a whole bunch of suburbs around, and then we're tracing the actual roads that are being used to trace of how this person would get to downtown. And we're seeing if this same principle applies. And we're seeing a lot of variation. Some cities are really, really close to optimal. Others are super far away. And so we're trying to make sense if this could be some way to actually constrain the structure of cities as well. But again, I don't have anything to show you about that right now, but it would be nice to see. I mean, in some, like, if you look at San Francisco, right, there's so many geographic constraints that you might not expect that the city would be so perfect because, you know, a plant can move anywhere in space. So I don't expect some of these cities that are so physically constrained to abide by this, but it'd be nice to see, you know, still.
Especially Miami.
Or Miami, yeah, I mean, so.
That's a comment. I think this work is really fascinating.
Thank you.
I mean, you said plants have been around for thousands of years. And we have been around for, well, I don't know how long. And we have a computer and some algorithms, and we think we've seen the light, that we are the messiahs, you know? But I think there's so much to learn from how biological entities have adapted and optimized on certain things that it will really open our eyes to what is to come for the future.
Thank you. Yeah, I mean, I mean, computer scientists generally agree with you, but I'm still struggling with the biologists to like get them on board with this type of thinking as a useful way for them to understand how their systems work. I mean, and it's getting better, but I agree that it's pretty fascinating. So, yeah.
So let me ask you. I look at the result, right? So I look at the result. So on day 4 or whatever it is that you're on the Pareto front at one point, right? That's sort of reflected 1 alpha. On day 20, it goes to another alpha. So I was just thinking about the trajectory and how actually it's moving along that Pareto front.
Yeah, it's a great point. We were trying to plot for an individual plant. X-axis is time, y-axis is the closest alpha on the Pareto front. And we were trying to see, is there any logic, like does it start off at some value and then go to some value? And it was just really noisy. We might need, like we only did this for about 20 plants. We might need a lot more to actually see the signal, but we couldn't make much sense of what the actual alpha values were how they were changing in a kind of logical way over time.
And also, I mean, the trajectory is always on the Pareto front, or is it actually a different trajectory?
Sorry, the trajectory.
I mean, say day 4 is here. Right?
Yeah.
Day 20 is somewhere.
You know,
right.
so it moves. So like you can see.
So it
like
moves along this front.
It moves along it, but it doesn't like always go this way or always go. It just we can't make sense of where it's going. It's just kind of, and some of it might just be just natural biological noise and randomness that it's using, or some of it might be some measurement error. Some of it might be, the soil content in one is a little bit different than the soil. There's just so many factors.
I just find that fascinating because so obviously you have one optimization. And then, of course, it's going to grow, and it's going to reflect that, and somehow it's retain that optimality.
So, I mean, in computer science, people have studied this online Steiner tree problem, where, like, you have a Steiner tree built on some points, and then you add a new point, and you want to add that point to your existing tree. But they've studied it in such a way that you can, and not in a way that looks at both of these objectives in a way that you can have this growth. But I think that's kind of what the plant is solving. Right, it keeps putting out new leaves, and it's growing. And so, how is it doing that? I don't know yet, but we have all the data online if you want to like look at plant scans, and that'd be great. But it's a cool problem. So.
Okay, only more question? Okay, then thanks. For the graduate student.